formestin is, again, a steroidal drug. Those are the compounds that we are looking at. And we have, of course, said how the enzyme works. Then we have the aminoglutathione. We have looked at the structure. This is a reversible synthetic competitive inhibitor. Its disadvantage is binding to various cytochrome P450 enzymes and inhibiting a range of steroid hydroxylation. So in other words, it's not specific. The problem of this one is that it is not specific for the aromatase enzyme. It is not specific for the aromatase enzyme. It will also inhibit other cytochrome P450 enzymes, and therefore, whenever you have a drug that is not selective for the receptor, then you find it will be responsible for a lot of toxicity that results in undesirable side effects. Then you have anastrozole and letrozole are more selective, and that's the advantage. Then for nitrogen of the triazole ring interacts with the heme ion of aromatase and provides binding of the steroid enzyme. So again, remember, they have, you look at this is the anastrozole, this is the nitrogen we are talking about. That nitrogen, of course, we also have that nitrogen there. So we're saying that is a nitrogen that's very important for binding the receptor. So we have seen the nitrogen for the triazole. So that's a triazole ring. You look at this, it has a triazole, it's 1, 2, 3, therefore triazole. So the nitrogen of the triazole ring is important to interact with the aromatase enzyme and provide binding of the steroid substrate. Remember, once the drug binds the enzyme and you get the drug enzyme complex, although reversibility means the drug will bind the enzyme a little bit more strongly, and as it binds it a little bit more strongly, it prevents the normal substrate from binding. It prevents the normal substrate, which is the androstenedione, from binding, and therefore the androstenedione is not converted to the estrone for the purposes of formation of the estrogen. So remember that when you talk about an antagonist is either reversible or irreversible. The irreversible ones almost bind permanently and destroy the receptor, be it an enzyme or whatever it is. But when you call about reversible ones bind a little bit more strongly. And when they bind a little bit more strongly, so within the time they are bound, the normal substrate cannot bind, and therefore they will bring some activity, though reversible. And of course, reversible competitive inhibition is usually concentration dependent. So as long as you have enough of the inhibitor, then the inhibition will happen. It's now when the concentration of the inhibitor goes down, that is when, of course, you get the reversal of the inhibition. Acts as a suicide substrate that permanently inactivates aromatase and is more selective in its action than aminoglutathione. So the formestin, which we also realized is a steroid. It's a steroidal derivative. This binds permanently. In other words, the formestin is an irreversible inhibitor. It's not a competitive inhibitor. It's an irreversible inhibitor of the aromatase enzyme. So this is our aminoglutathione. Let's look at this structure. And if you look at that structure, you'll be able to see how it is metabolized. We are saying most of it, about 50% of this drug is excreted and changed, because you can see whenever you have an amino group, it means that can be protonated at physiologic, although this is a bit hard to protonate because of that, but this one here, even this one is a bit hard because this is electron withdrawing that electron withdrawing. But nevertheless, the whole of this group here, and of course that amino group, are bringing in polarity into this molecule. So you find this drug, quite a lot of it can be excreted and changed. About 50% of what that means is excreted and changed in urine, but it can also be metabolized to the N-acetyl aminoglutathione. N-acetyl, which means this group can undergo acetylation. as N-acetyl aminoglutathione. That means our aniline nitrogen undergoes acetylation, and if that happens, you get N-acetyl aminoglutathione. Then you can also have a metabolite like hydroxyl aminoglutathione. Hydroxyl aminoglutathione. Where would we get the hydroxyl group? And then we can also get two minor metabolites like para-nitroglutathione. This group here again, I'll think of the amino. This group here again can undergo oxidation. That group there can undergo oxidation to the nitro group to give us the para-nitroglutathione. Then you can also have the formyl aminoglutathione. Formyl aminoglutathione. Which group can undergo? This group there can undergo oxidation to formyl aminoglutathione. Because when I look, the only group that can undergo oxidation is this group here, methyl. Remember, it can undergo oxidation first. Oxidation will go to the alcohol, and then from the alcohol it can go to the formyl group, to the aldehyde, and then to the carboxylic group. So I'm suspecting this is the only one. I can't see any other group within this compound that can form a formyl. So I'm suspecting that this is a group that can be oxidized to give us the formyl aminoglutathione, because it can go to form carboxylic acid, and sometimes after forming carboxylic acid, the carboxylic acid is cleaved such that you have this carbon here, this carbon here comes CHO. That will be the formyl aminoglutathione. I don't think there's any other way you can form a formyl, because it cannot undergo methylation. This is the only one which can undergo formation of a formyl group. And then there was N-acetyl. Where can we get hydroxyl amino? Perhaps there's still this group here, but I'll confirm. But I believe the only other group that can undergo also hydroxylation is this group there. That methyl group can undergo and form CH2OH. I think that's what they are calling hydroxyl glutathione. Hydroxyl glutathione. Again, this group, this methyl group, undergoing oxidation. First of all, remember, whenever it undergoes oxidation, first of all, it goes oxidation to the alcohol. So once it undergoes oxidation to the alcohol, it will give you hydroxy aminoglutathione, because it's only that group that has changed into hydroxy group. And as we have said, I believe this group can be also converted in, it's first of all an alcohol. The alcohol is usually changed into a formaldehyde and then into a carboxylic acid. So that alcohol can change into a formaldehyde. And once that alcohol changes into a formaldehyde, it will give us the formyl aminoglutathione. And we have looked at the nitroglutathione and so on. So those, they are very simple routes of metabolism, mainly because this drug already has very good water solubility. It also has the primary. Remember, it has some of the groups which you require to add for phase 1. It already has the functional groups that are required for secondary metabolism, but doesn't seem to undergo any secondary metabolism. It's mainly undergoing primary hydroxylation there, oxidation there, so it is not undergoing any conjugation. The next one is the 5-alpha reductase inhibitors, and we have finasteride and dutasteride. And the finasteride, again, you can see it's 5-alpha reductase inhibitors. Let us look at first the role of the 5-alpha reductase enzyme, which is NADPH dependent. Again, the testosterone undergoes reduction to 5-alpha, to dihydrotestosterone. Dihydrotestosterone, you look at this double bond there. Remember, reduction also involves the addition of hydrogens, so it undergoes reduction there. So you can see this does not have the hydrogens there, catalyzed by the enzyme 5-alpha reductase enzyme, which is NADPH dependent. So that is a dihydrotestosterone, and the enzyme is very, very important. The enzyme again is 5-alpha reductase. You can see the structure of finasteride. So finasteride again is a steroidal drug, and therefore it has structural resemblance with the normal substrate and taken up by the normal substrate and by the enzyme in place of the normal substrate. So we are saying 5-alpha reductase inhibitors will prevent the conversion of testosterone to 5-alpha or to dihydrotestosterone. So we have finasteride. It's orally active. It reduces the levels of dihydrotestosterone. It is used in benign prostate hyperplasia, so it will reduce the prostate volume. It will reduce the symptoms. It will peak urine flow. It will increase the peak urine flow. It will reduce, of course, the dihydrotestosterone levels. Of course, when you have prostate cancer, whether benign or malignant, the major problem is that because of the growth volume of the prostate, it brings blockage, it squeezes the urethra, and squeezing the urethra, it might bring a stricture, and therefore urine doesn't flow, and that is usually one of the terrible symptoms, that the person cannot even be able to pass urine, and that is quite uncomfortable for the patient. So we are saying when you give finasteride, it will help in shrinking the volume of the swollen prostate, and therefore, of course, relieve the urine blockade of the passage of urine blockade, and therefore help the person in terms of relieving the symptoms. Finasteride is an azasteroid, in that you can't see it has an NH group there. So we are calling it an azasteroid. That's a synthetic drug for the treatment of benign prostatic hyperplasia, but it's synthetic, but of course it does have steroidal structure. It has a law as an androgen antagonist. It blocks the action of the enzyme called 5-alpha reductase, testosterone reductase, 5-alpha reductase. This enzyme changes testosterone to dihydrotestosterone that causes a prostate to grow or hair loss in the males. In other words, the dihydrotestosterone is the one which is responsible for the tumor growth. So once you remove it, then you will stop the tumor growth. It will shrink. Again, most of these are just to alleviate the symptoms, of course, not to cure, but usually for you to do proper treatment, you must do some surgical intervention. But all we are saying is to shrink the prostate. The person at least will be able to pass the urine. The discomfort, most of the discomfort will be alleviated. It will increase testosterone levels in the body, which decreases prostate size and increases hair growth on the scalp. So that, again, is the 5-alpha reductase inhibitors, again also used in the management of prostate cancer, because it will reduce, as we have said, dihydrotestosterone. Again, for such patients, they will of course also have a lot of side effects. There will be quite a number of side effects, because it's like you are decreasing their masculinity. It's like you're decreasing their masculinity, because once you reduce the levels of testosterone, it's like you're reducing the masculinity of that person. And that's why some of these drugs, their compliance is usually very low, because of the side effects that the patients will encounter. Then we also have the luteinizing hormone, releasing hormone agonists. Remember, we have what we are calling the luteinizing hormone. It must be released. And once you release the luteinizing hormone, it will go and act on the gonads now to release the androgens or to release the estrogen. So we are talking about now the luteinizing hormone releasing hormone, the luteinizing hormone releasing hormone agonists. Examples are buserelin, gonadorelin, goserelin, histrelin, leuprolin, and so on. So all those are drugs that can be used to block the luteinizing hormone releasing hormone. Because remember, you have a luteinizing hormone releasing hormone which is produced in the brain. It's produced in the brain. It comes, I think, into the hypothalamus, if I'm not wrong, and then triggers again now the release of the luteinizing hormone. So we are therefore saying the luteinizing hormone, releasing hormone, also known as gonadotrophin releasing hormone, is released by the hypothalamus and stimulates production of luteinizing hormone from the anterior pituitary. So remember that arrangement. You have the hypothalamus. It will release the releasing hormone, which is a luteinizing hormone releasing hormone from the hypothalamus. The luteinizing hormone releasing hormone from the hypothalamus comes into the pituitary, triggers the pituitary now to produce the releasing hormone, the luteinizing hormone. That's why we are calling it the luteinizing hormone releasing hormone, because it's the one which first of all is produced, goes to the pituitary, and triggers now the production of the luteinizing hormone. So if you block the release of the luteinizing hormone releasing hormone, then if you block the release of the luteinizing hormone releasing hormone, then it means you will not release the luteinizing hormone ultimately from the anterior pituitary. Once you do not do so, then of course again you will not release your sex hormones. In response to luteinizing hormone, testosterone is produced by the Leydig cells in the testes. Luteinizing hormone releasing hormone is a decapeptide hormone which binds receptors on the anterior pituitary cells and stimulates the release of the luteinizing hormone. Decapeptide means it has 10 amino acids. So this luteinizing hormone releasing hormone is one which has 10 amino acids. You release it first, as we have said, from the hypothalamus, goes to the pituitary, then triggers the release of the luteinizing hormone. So if you can get now, because it's known to be a decapeptide, you can synthesize, or you can have other drugs, or most of these drugs are actually amino acids, or they are like proteins, or they are decaproteins or decapeptides. They are decapeptides that have structural resemblance with the luteinizing hormone releasing hormone, but they can bind more strongly and therefore bring about antagonist activity. On long-term exposure of a luteinizing hormone releasing hormone, the receptor becomes desensitized, leading to a drop in luteinizing hormone levels. Again, this we have said severally, that if you continue exciting any receptor, you excite it repeatedly, you continue exciting that receptor, then it will get to a point and it will get receptor fatigue. To get receptor fatigue, it will not be able to produce what you want. So remember, these are agonists. And because they are agonists, they will go to the luteinizing hormone, they will go to the receptor they will go to the receptor within the hypothalamus, which is responsible for producing the luteinizing hormone, releasing hormone, and bind on it, and bring about fatigue, so that the luteinizing hormone, releasing hormone, is not released. And once, of course, you do not release it, then for sure, then again, you will have low levels of the testosterone. Since luteinizing hormone stimulates the synthesis of testosterone, this results in lowered testosterone levels. So we are overstimulating. We are giving agonists which are overstimulating the receptor within the hypothalamus, and therefore that receptor is not able to produce a luteinizing hormone, releasing hormone. Once you don't produce that hormone, then you will not produce luteinizing hormone. You don't produce luteinizing hormone, you will block the production of your androgens. So we are saying, like an example of the drugs, which is leuprolide and goserelin, these two are both decapeptides analogs of LHRH, designed to be more resistant to peptidase degradation. So they have more activity. They are not readily degraded. So because they are not readily degraded, they have structural resemblance with the LHRH, have longer duration of activity, and therefore they will overstimulate the receptor and render it inactive. And if you look at them, so this is the LHRH, you can look at this and you can see at position number 6, you have a glycine, and at position number 10, you have also a glycine for LHRH. So this is the luteinizing hormone. So when you have the drug like leuprolide, you bring in an unnatural amino acid D-leucine, and then in place of glycine, you put another group, an ethylamide, and that allows stabilizes, makes that drug stable, allows that drug to bind more firmly, and therefore overstimulates the receptor, giving us the activity we have already talked about. And then you look at goserelin, and again, if you look at goserelin, you can see all up to there, the molecules are the same. And again, at position number 6, again, you give a D-tart butyl serine. D-tart butyl serine, you can see now the glycine has been replaced with D-tart butyl serine. And then you can see this leucine, arginine, proline. These ones again are the same. And again, you put a different molecule at that position. And again, once you do that, this compound has better binding, is more stable, and therefore it will overstimulate the receptor and finally give what we call receptor apathy. And once you get the receptor apathy, then there will not be production of the luteinizing hormone and hence no activity. So glycine at position 6 is replaced with a natural amino acid, making this region unrecognizable to the metabolizing enzyme, and therefore giving leuprolide a much longer duration of action. Also, substitution of the glycine residue at position number 10 with a suitable group also increases receptor affinity. So you have both increased receptor affinity, you have decreased in metabolism. That molecule has a much better activity at the receptor, overstimulates the receptor, results in receptor apathy, and therefore no activity. The final group I have, but I'll still look for, I think there is one group we have not discussed. So the final group of drugs that, I think this is not here. That's not a hormone. So we will, first of all, we were looking at the hormone therapy. So for hormone therapy, we will end at luteinizing hormone-releasing hormone agonists. There might be others I've not talked about, but as I said, you cannot be able to discuss all this. There are too many. You can't discuss all of them, but you try the best you can to give enough examples. Is there any question? Have you handed in the assignment? Frida, have you collected the assignment? Frida, have you collected the assignment? Frida, are you there? Yes. So if we have just briefly then, because of time, then we can first go back and just introduce very briefly, because time is never on our side. I don't know where time goes. Every time there is no time. And wonders where time goes, really. So I had left out the alkylating agents because of your assignment, and if the assignment is ready, then you can just do something. So we are now done with the metabolites. We are done with the plant metabolites, plant products, the hormone therapy. So then the final one we look at is the miscellaneous. And the interference. So we are looking, I want us now just briefly to introduce the alkylating agents. And for us, of course, to understand the alkylative agent, we must understand the DNA, how the DNA structure. And of course, as we had said earlier, our DNA has a backbone, as you can see, of a phosphate and a sugar. That backbone. And also on this side, it will have the same backbone for the DNA helix, where you have 2 strands. And then, of course, in the middle, you can see in the middle, you have the bases. So on the same, this will be repeated on this other side, so that at the end here, there will be a base, and this base will interact with that base through hydrogen bonds or through hydrophobic bonds. And this is where we were saying that you must have, on this side, you must have a certain base to interact with the base on the other side. So there is a particular pairing whereby, of course, we have the GC guanine pairing with cytosine. If you change that pairing, then you destroy, definitely, you will have you destroy the DNA, and of course it will not function. So that's the first important thing, is for us to understand the kind of structure, the DNA structure, and that DNA structure must be maintained for the DNA, especially for the DNA to replicate. If you change that structure, whereby you have said you must have certain base pairs or certain purines at certain specific purine pairing with a specific pyrimidine, and then of course you have that arrangement where you have a backbone of the phosphate and the ribose sugar, another backbone of the phosphate and the ribose sugar, with your bases in the middle. Change that structure, then you will not allow for replication, and you will not allow for the separation, because you start with a DNA helix where you have 2 strands, and for you to have replication, transcription, so that you can have transcription and so on, you must separate the strands. The strands must be separated, and one of them is what will act as the template, either for protein synthesis or for whatever you want to synthesize, or for DNA synthesis. This, of course, it's important for us to know which is which. When we look at adenine, it has an amino group at position number 6. Guanine, at position number 6, it has an oxo group and an amino group at position number 2. And of course, the adenine and guanine are purines. And then, of course, we have thymine. Thymine, of course, is a derivative of uracil, where the position 5 has, of course, a methyl group. And most of these things we had already mentioned, but it's important for us to remember, because as we can see, alkylation of DNA, of the bases of the DNA, occurs at particular points. So it's important to understand the structure of each of the bases, and of course, which points are more susceptible to the alkylation. So we are looking at alkylating agents. Alkylating agents are highly electrophilic compounds. In other words, they are electron deficient, that react with the nucleophilic groups within the DNA. And some of the points of nucleophilicity within the DNA is where you have these nitrogens. Remember, nitrogens usually have lone pairs of electrons. And because of the presence of the lone pair of electrons, or even oxygens, they will have lone pairs of electrons. Because of the presence of the lone pairs of electrons, then you have electron excess, and therefore nucleophilic centers, and react with nucleophilic groups on the DNA to form strong covalent bonds. Nucleophilic groups on the DNA bases include, so you have the guanine, where you have N2, N3, and N7. If you look at guanine, this is our guanine. N2, that's position 1. Position 2, you have this group there. N2, N3, and N7. This is N7 there. This usually is what binds. That is what binds with your ribose sugar, so it's not available. So we are talking about N2, N3, and N7. So those are positions that are available for alkylation. For adenine, we have N1, N3, N7. Again, N1, N3, and N7. So both of them have the N7. And indeed, for guanine, the most susceptible position is actually N7 for guanine. And guanine is the one which is most susceptible, I believe, also because of the presence of this amino group, which is an electron pusher. So it will push electrons into the system. And because of that, you find that guanine is the one with highest susceptibility of alkylation, especially at position number 7. For thymine, cytosine, and uracil, you can also note for thymine, it's oxygen position 6, and for cytosine, N3. So for cytosine is usually N3. So position 1, position 2, position 3, N3. And for uracil or thymine is N6. 1, 2, 3, 4. Position 1, 2, 3, 4, 5, 6. Oxygen 6. So for thymine, usually the oxygen 6, and for cytosine, N3. So those we are saying those are the areas that are more likely. The others, but those are the ones which have the higher susceptibility. But as I have said, the one with the highest susceptibility of alkylation is guanine N7. And you find that most drugs indeed will alkylate guanine N7. For many alkylating agents is N7 position of guanine, whose nucleophilicity may be enhanced by adjacent guanine residue. In other words, if you have adjacent guanine residues, then again the nucleophilicity of N7 is even further enhanced. So what are we saying? You look at adenine, we have said position 1, 3, 7. So we are saying these are the nucleophilic groups, as we have said. Guanine, we have said N7 mostly. N7 high susceptibility. Cytosine, we have already said position. So those are some of the positions that can undergo alkylation. Now, when you talk about an alkylation, remember what is coming in is an electrophilic group. So it will come in with deficiency of electrons, and therefore once it comes in, it can alter the shape. It can alter the tautomerism. It can alter the distribution of electrons within that molecule. If it alters the distribution of electrons in that molecule, it can lead to a number of things happening to the DNA where it has bound. So it comes with an electron deficiency. It has bound on the base of one of the bases, especially the guanine base. And in binding the guanine base, or let's say the nitrogen 7, it has come with electron deficiency. It will have an influence on the distribution of electrons within that base, and therefore hence within the whole DNA, and it can lead to quite a number of effects, which will be mentioned one by one. Alkylation, some of the effects, alkylation converts the base to an effective living group, so that attack by water leads to depurination and the loss of genetic information. That is just what we're saying here. So if you come and attack that group there, this is guanine. So you can see it has come in with electron deficiency. So this position there becomes positive. And it has an influence on the whole molecule. It makes it unstable. It makes this molecule very unstable. And therefore, because there is always water, it will lead to depurination. So there will be a cleave there. And that and its phosphate will be released. And of course, you will release. You are releasing it. The purine, of course, is already an alkylated purine. So you're not using it as a purine that can be used. It cannot be used. It's already alkylated. And therefore, since this was part of the DNA, it's carrying DNA material, it's carrying DNA information, and that DNA information is going to be lost. And therefore, that will that will interfere with the synthesis of other DNA molecules. Alkylation has also been proposed result in altered base pairing away from the normal. See, normally you have GC binding. So you find GC and, of course, adenine thymine also binding. So once you do alkylation, as I have said, there's a change in the molecule. And that change in the molecule now changes the pairing, so that you might find the guanine now binding to thymine instead of binding to cytosine. And that, of course, difference in base pairing again will mess the DNA replication. So instead of GC80 hydrogen bonds, where from the normal GC80 hydrogen bonds, because of alterations in the tautomerism. In other words, once again you bring this molecule, it has deficiency of electrons. It will have a change in the distribution of electrons within these spaces, and the tautomerism, and therefore have an influence in the base pairing. Once you have the long base pairing, those bonds might be much stronger. Those bonds will influence the DNA and therefore also influence its replication. Alkylation also leads to increased acidity of the N1 nitrogen, reducing the pKa from 9 to 7. Once you do alkylation, when you do an alkylation, that means you are taking away the base, the nitrogen base is giving away its electrons, so it's becoming more acidic. The molecule is becoming more acidic, and that's why we are saying alkylation also leads to increased acidity of the N1 nitrogen. In being acidic, we are saying it has been alkylated. For it to be alkylated, it must give away its electrons. Once it donates its electrons, that means it will be a weaker base. Once it's a weaker base, it means it's a slightly stronger acid, reducing the pKa from 9 to 7 or to 8, giving rise to azionic form that may also mispair. So all we are saying is that now that molecule can acquire, that molecule that has been alkylated can acquire a positive charge, and we can have a zwitterionic form, and that can also mispair. For those agents that possess two reactive functionalities, both interstrand and intrastrand cross-linking becomes possible. When we look at some of these drugs, we will find that some of them have can be able, one drug can alkylate either, it can alkylate either the different strands. One portion of the drug alkylates, let's say, strand A, and the other portion of the drug alkylates strand B. Remember, we have two strands. This drug intercalates, goes in the middle. One strand is able to alkylate strand A. The other drug alkylates strand B. That means there cannot be any separation. That is what we call the interstrand alkylation. It can also alkylate within the same strand. Let's say there is a guanine at position 10, and there's a guanine at position number 8. One strand can alkylate the guanine at position number 8, and another one alkylates the guanine at position number 10 or 11. That is what we call the intra-strand. So you can have either interstrand, where two different strands of the double helix DNA are alkylated, and therefore there cannot be separation. Or you can have whereby you have intrastrand, where different bases within the same DNA strand are also alkylated. And of course, as we have said, we'll have an influence on that, the type on those bases, on influence of the DNA, and therefore have an ultimate influence on the transcription or the replication. When intrastrand links occur, separation of the two strands during replication is prevented, and therefore replication is completely blocked. So you can have those two happening, interstrand or intrastrand. Strand inter, of course, will bind the two, or will bond the two different strands. They cannot separate, they will not allow for replication. When you have the intrastrand within the same strand, it will also have an influence. It might bring about depurination, it might bring different effects on that DNA, and therefore, again, there will be a negative influence on the replication. So this is what we are talking about. This is the inter. So you can see this is a guanine on one of the strands. This is one of the strands going down. And then this is another guanine on another strand. So you can see this is a drug. When you look at the nitrogen mustards, you see that usually they have two alkylating strands, and what differs in most of them is the R groups. And I hope you are able to look at that. That what differs in them is the R group. Like if you have like drugs like mephalan, you have drugs like chlorabusine, you find that what differs is the R group, but they have two strands. They have two active chloroethyl moieties, which can actually alkylate. So once you do this, it is completely held together, and therefore it cannot be able to separate. So this is again what we have said. So that reaction there, giving it positive charge there. Once it is positive charge there, then again with water, there can be depurination. So you can see that can happen. Also, once you add water, there can be a cleave here. And you can see this is what happens. So there is a clip there. So there's destruction again of information. Or with this again here, remember we said there can be sweet ionic formation. Here there can be bring a bit again a change in electron arrangement, so that now this oxygen is converted into a hydroxyl group. You can see now this was a dihydro derivative, dihydropyrimidine. So if you look at it here now, it has become a proper pyrimidine, whereby it now has a hydroxyl substituent. And if this hydroxyl group, remember hydroxyl group phenols can undergo minimal ionization, very minimal ionization. So if you get that ionized and you also have this, you will have what we call the zwitterionic position, where in one group you have a hydroxyl group and you have a negative charge, and of course in the other group you do have the positive charge, and therefore sweet ionic groups. And of course also, not only do we have the sweet ionic groups, remember when you change this group to a hydroxyl, from an oxo to a hydroxyl group, you change the ability of this compound to bind. It cannot bind now. It is not able to bind the same way as it was binding when it was an oxygen. So most probably it will just leave it will bind with another group, or it will bind with another part of the DNA. And when that happens again, that will have an influence on the replication. Because remember that this molecule of guanine is being able to pair with cytosine because of its structure the way it is. Once you change its structure, then you find that it is not able to pair through the hydrogen bonds. It will not be able to pair with cytosine. Again, you will alter, as we had said, you will alter the pairing. And once you alter the states pairing, again, you will interfere with DNA replication. Of course, finally, also deformation. So there can be, so once you have this, so this was a minus here, it can, this minus, so this has become a double bond. So this oxygen here, when it is a minus, because it can undergo ionization, we first remove it so that I can be able to show you properly. So remember, when it's a negative, a lone pair of electrons here can move in here, that position, and that will become a double bond. That will become a double bond. Once that becomes a double bond, these electrons will move to the nitrogen and it becomes a negative. Again, you can see within that molecule creation again of the zwitterionic molecule, and again having a major influence on the pairing of the initial guanine with the cytosine. So these are the various changes, the various effects alkylation can have on the DNA. Alkylation can have several effects on the DNA, including depurination, also cleavage of the pyrazolone. This is a pyrazolone. Yeah, pyrazo, sorry. So cleavage of the pyrazole group. This is a pyrazole group. It can undergo cleavage, as you can see here. It has cleaved to give us an amide there. So there can be cleavage. Of course, once it cleaves, of course, there will be destruction of the DNA. There can be change of the substitution within the pyrimidine ring. Again, that change in substitution will also have an influence on the bonding. And of course, also finally, you can also have what we are calling there zwitterionic development. And once you have zwitterionic development, then again, the way the molecules will bind the bases with the base, the resultant base will bind with the other bases will be completely different. And once that happens, then there will be a big influence on transcription. And once you cannot have replication, especially if it is happening in the abnormal cells, then of course you will stop the tumor growth, because that's what we are trying to do. you will stop the tumor growth, and therefore you will have, the drug will have done what's supposed to do. And in that class, of course, we have different drugs. As we have said, the general structure of the first category of drugs, which are alkylating drugs, which are the nitrogen mustards, is where you have an R there, and then adjoined to nitrogen. And then from the nitrogen, you have the ethylene, chloroethylene groups. And this is what is changing. So if you look at methylene, you can see that is the same there. And then R is a phenylalanine. You can see this is phenylalanine, and that gives us methylene. Then you can have also the uracil mustard. You can see that is the same portion there. These are R, which is uracil. Then you can have estramustine, and again, you can see our R, which is up to there. Of course, this is an estrogen. That's why we are calling it estra, as we have just discussed. That is an aromatic group. So it's an estrogen ester, and that is our R, and that is the same. You have chlorabusel, again, maintaining the same there, and this is our R. And if you look at our R, it's phenyl, 1, 2, 3, 4, phenyl butanoic acid, and that gives us chlorambusel. And then you have ifosfamide. Ifosfamide, again, you can see this is supposed to be a phosphate, but mide, it's a phosphate. And of course, we have the ifosfamide and the cyclophosphamide, whereby for the cyclophosphamide, this will not be there. That will not be there, and the phosphate here will be joined to 2 of this. So you have another one like this one here, and you remove this. So this will move from there, and it will, let me remove it, let me do it better. So for cyclophosphamide, the whole of this group here will be shifted, and it will come there, that phosphate. So if you look at your structure of cyclophosphamide, this P has 2. This P, I hope that's the correct thing. So let's just confirm. I know I have the structure of cyclophosphamide somewhere. Yes. So you can see when you look at cyclophosphamide, in ifosfamide, this N, so the hydrogen here is replaced with one of these. So you have that one there and this one there. But for cyclophosphamide, you can see the 2 groups. There is a P and then the nitrogen, and the 2 groups are coming from the phosphorus. Unlike in the ifosfamide, one of these group here comes from here, ifosfamide. So, but for cyclophosphamide, the 2 groups are attached to the phosphate or to the phosphorus atom. So those are the examples. And when you look at them easily, they are not so hard to remember, because as I have said, of course, there's clomethamine, or meclomethamine or clomethine, when R is a methyl group. Methylene is when your R is a phenylalanine. Uracil mustard, that's easy, when it's an uracil group. Estramustine, it's an estrogen ester. Chlorabusel, it is a phenyl butanoic acid. Ifosfamide, that's the only one you must get yourself to have a way to remember. So indeed, this group was coming from here, that you have been here from here. So this hydrogen has been replaced by another group. So the phosphate carries the 2 groups, and unlike in ifosfamide, where you have the 2 different nitrogen, each of them is substituted. So please learn these structures. I know you have a lot to learn, but try to learn these structures, because we are going to look at one by one. I mean, we are going to look at them. And since they have a general structure, it's very easy to discuss them, because once you have understood the mode of action of one, it's, of course, with the exception of a few details, you will understand the mode of action of all of them, since indeed they do share in their general structure, whereby it is only the R. For the different drugs, it is only the R which is changing, but otherwise, and of course, there is a reason why R is changing. R is changing to make the drug more stable, because like chlormethamine or meclomethamine is very reactive, and it will form the aziridinium ion, it will form that ion within the tissues, and therefore bring about problems within the tissues. When you stab, and of course, it will not be given orally. But now when you put the bigger group, especially with the phenyl group, you stabilize the molecule, you give it time, it becomes more selective, and it can wait until it gets the receptor for it to be converted to the active moiety, and then now active moiety, you bind the tumor, because the active receptor, the active site is a tumor, so it can wait to get into the tumor and bring about its activity of destroying the DNA within the tumor. But when you give it like chlormethamine or meclomethamine, then it's too reactive, and therefore it will bring its activity. It will destroy DNA everywhere, and it will also not, and therefore it will not have any oral activity. I think with that, unless if I believe there are people who join very late. Now you are 42. Where were you? You join the last minute. I will discover who you are. So there are those who join very, very late. I don't know where they come from. Remember, it is for your own good. Mimi, see me, Mimi. So do not join the last minute to deceive me that you are there. It's for your own benefit. So do it for yourself, not for me. So I think with that, at least we have, I'm hoping that I had asked for an extra lesson next week. So I think we make it for anti-cancer, so that now we should be able to finish the anti-cancers next week. If we can't finish the anti-cancers next week, we can also have a cut the following week or so. You have said that you want your cut for the narcotic analgesics on 25th. We are really pushing these cuts, but anyway, prepare well. Let's then have the cut on 25th. It will be a very simple cut. I think only 2 questions. Very, very simple cut. So start reading also on the anti-cancers. If we could have, as I've said, an extra lesson next week, I believe we will summarize on the anti-cancers. We still have the antivirus. At least this topic is a little bit better. It has only 2 topics. It has only 2 subtopics, unlike the narcotic one, which has several. I was asking for 2 lessons, but I think class rep for now said we can have 1. So I will take it kindly. If you can be able to organize for another one from next week, such that you can have 2. If we finish early, the better, for your own benefit, for you to be able to revise early enough. Any questions? So can you sign in then?